Okay, good day, everyone. Welcome to our webinar. Today, I will introduce my experience in developing the toolkit Spateo that is designed for the spatial transcriptomic analysis, especially for the multi-time points, or we call that developmental stages. So this is a topic about how to spatial temporal modeling of a virtual embryo. Okay, before my formal presentation, I will give my own understanding of how to define a good algorithm in the biological field. Here are two small stories shared with you. First one is one of my colleagues once asked me about the populational analysis of bacteria. He said he had already used the most widely used and the gold standard pipeline. Sorry, wait for a moment here. Yeah, GATK to analysis the bacterial populations. However, he found that the result is weird and he could never get the correct answer as he thought. So I just asked him a very simple question. For the GTK is designed for human genome. All of us know that the human genome is a diploid genome. For the gene types, we on the genomic locus, we have altogether 10 gene types, including the homozygous AATT-GGCC and other 6 types of the heterozygous like AT, AG, AC, and others. However, for bacterial population, most of bacteria only have the monoploid, and they only have 4 types of the gene types. So we use a diploid modeling to get the population analysis for bacteria. The concept is wrong, so he could never get the right answer for this analysis. And another interesting story is a very, very simple algorithm, but published on Science and has get more than 1,000 citations in recent 5 years. This software called Cytotrace. Cytotrace is used to analyze the developmental potential of the cells, or we call that the stemness of cells in single cell on data. They found that the gene count within one cell is highly positively correlated to the developmental potential. So all of the algorithm is only to calculate the correlation between the gene numbers and the potential of the development. It even cannot be said as an algorithm because to calculate the positive correlation is too easy for every bioinformatics analyzer to do that. However, they found this universal phenomenon in different data sets, and it is a very robust algorithm to study the single cell on data. So it gets very high citations in such short time period. So these two stories reflect a very basic concept in developing the algorithm in biological field. The innovation of the algorithm itself is not that important. The more important thing is the innovation of concept and the robustness as well as the generalization of the algorithm to fit for the diverse biological scenarios. So in our Spatio toolkit, we also use this concept to develop the algorithms. And many many people ask me about why the spatial transplants become so complicated to analysis, because there are already a large amount of well-performed single-cell RNA data analysis toolkit or software in this field. It seems that the spatial transplants only increase the geographic location for these cells. Why the analysis become so complicated, only increase the coordinates for the cells. So let me try to explain the reason. We will begin from the bulk RNA data analysis. For most bulk RNA data analysis, we only have around 10 samples, 10 biological replicates for analysis. And actually, we need to analyze all the genes that comprising 20 to 30 dimensions of the data. However, most of algorithms just simplified this calculation into 20 to 30,000 genes, and each gene has only one dimension. So for one-dimensional analysis, 10 replicates would be sufficient to get robust statistics. When we go to the single-cell RNA field, now the cell number increased a lot. We have thousands, tens of thousands, even hundreds of thousands cells. This analysis becomes a populational analysis. Notice that when we comparing the different gene expression between two cell types, it actually compares the populational differences. So for each population, we have many biological replicates, or we call cells, and the comparison is based on what I said, 2,000 or 3,000 dimensional data. So this analysis, because we have more samples, becomes robust. However, for the spatial transplants, we aim to analyze a target cell, while the neighboring cells, how these cells communicated each other is a research hotspot in this field. So it is not for populational analysis. It is a niche analysis, or we call it microenvironment analysis, for one target cell and its neighborhoods. There are not enough biological replicates to demonstrate your algorithm. So the spatial transplants becomes quite complicated to analysis. Thus, we need to have sufficient data to study the spatial transplant. A 3D regeneration of mouse embryo development is a very good case study in this field. As we can see, for current available 3D mouse embryos, from the embryo implementation to even adult, we have plenty of data to construct our algorithms and demonstrate our algorithms. So Spateo is based on such kind of data, and it used diverse multidiscipline algorithms to analyze the spatial transplant data. It includes like physics, geographies, and even economics methods. We implement these methods into spatial transplant data and fit for our biological scenarios. So here I just briefly introduce the data used in Spateo paper. We have both 2 time points of mouse embryos and Drosophila embryos. For the mouse embryos, we focus on 2 main organs. One is heart and another is CNS. The heart is the first developed organ in mouse embryo. So the relative cell proportion seems decrease in the comparison of E9.5 and 11.5 mouse embryo. However, the number of cells increase. In comparison, the central nervous system is intensively increase the cell number during this stage. So we just select heart and CNS as a demonstration of Spateo. For all the algorithms, as I said, we just study we just study the biological phenomenon, and then based on these biological events, we abstract the models from the detailed events and try to solve more biological problems based on the abstract model. So let me introduce the detailed algorithms. First one is the algorithm in three-dimensional reconstruction. For the previous methods, most methods are derived from the image alignment. When we register two images, in image field, we usually use global alignment that aiming to register two images globally. However, this is not the case in biological events. We know that in real biological events, especially during development, cells can be apoptosis, and also it can be migrate to some other place. All the cells can be divided or differentiate to more than one cells. So in the algorithm field, it can be represent 1-to-0 alignment, 1-to-1 alignment, but the place of the cell is changed, and 1-to-multiple cell, 1-to-many alignment. So the registration process or the alignment process can be changed to a generative process. Considering that we have a slide A, and the cells, each cells in slide A can be generated to another, together generate to another slide, which is quite similar to slide B, but for each cell, the cell can undergo apoptosis and division to 0 or many cells. So the generative process is a kind of local alignment for each cell, and they all together to generate a new slice quite similar to slide B. So we just change an alignment algorithm to a generative process. So why this alternation is useful to interpret biological events? Here is a very good example. For this stage of the central nervous system, the neural tube is still a hollow tube. And when we sequentially cut the embryo into sections, we will have the cell wall of the tube in the light blue, and we also get the lumen, the hollow tube, the lumen of the neural tube. So these two parts, although they are physically closed, they cannot be aligned together. In many public algorithms, because they need to achieve the global alignment, so the alignment seems to be rotated to have a minimal loss for that global alignment. However, we just consider about the cell apoptosis, so the neural tube itself can be generated with different cell numbers and the topology, the shape of the neural tube can be quite different. While for the other parts, we still get the well alignment result. So in this scenario, the local alignment seems more biological reasonable than global alignment. So we just use the generative process to do the 3D reconstructions. And here is another example. The no biological event is during the development. For example, in Drosophila, many genes have the gradient expression along the developmental axis. We call this phenomenon in biology is patterning or regionalization. The genes, for example, one gene could be highly expressed in anterior, but it decreases along the axis to posterior. So we just abstract the model into potential field. This is an example for potential field. I use the contour line, the SL height, to demonstrate this method. So for such development issue, to find the genes that have gradient expression, or it can perpendicular cross the iso height, is the genes that of our interest. This development phenomenon is widespread in different tissues. For example, in cortex, we have different layers as well as the different functional columns in the cortex. Even within a cell, with the distance to nucleus and to cell member, genes also have spatial gradient along this axis. In the real biological scenario, you can see the cortex. For example, this is a macro cortex. It's quite complicated comparing the cortex we demonstrated in mouse. So the algorithm is designed to solve the boundary problem in this true biological scenario, and we result many gradient gene expressions in our developmental result. The third one in our paper is cell-cell communication algorithms. These algorithms are highly developed in spatial transference. So I will not only introduce our algorithms, I will systematically introduce all the algorithms, or I call that the algorithm development in spatial transcriptomes. In single cell on data analysis, as we lost the spatial coordinates for the cells, we only calculate the co-upregulation of the ligand and the receptor in single cell on data. However, when we face to the spatial transcriptome data, we can advance our method to calculate the pleiotropic effects of ligand and receptors. It means that one ligand can match to more than one receptors, or one receptor can match back to several ligands. In such algorithm, we will find the more weighted ligand receptor pairs comparing to other pairs. So in Spatio, we advance this concept into inferring all regulatory networks. For the target cell, we just predict all the expression pattern of this cell, or we call that cell profiling, or the RNA profiling of this cell, by calculating the neighboring cells and the ligand receptor weights for these cells, as well as calculating the downstream TFs and the target genes. So when we construct a network, we can infer all weights, not only for the ligand receptors, as well as the downstream TFs and the effect genes. So if this is true, a very new concept can be constructed. This method I read from bioRxiv recently, called Luna. It is an AI model. It predicts all the spatial arrangement for the single cells. For example, we don't have the spatial transfer data. However, we have already proved that for a microenvironment, the target cells, the expression of target cell can be inferred by the neighboring cells. So the whole spatial arrangement of cells might be inferred by solely single cell on data without any coordinates. This is a very interesting topic, and I think in near future, many algorithms in spatial transforms can be derived from this concept, especially for the neighboring cell communications. And here is a demonstration of the previous methods in Spateo. We focused on organizer called ZLI organizer during the brain development. The ZLI organizer is a physical boundary in the dicephalon. It separates the P2 region and the P3 region for dicephalon. And later on, P3 region will develop into pre-thalamus, and the P2 region will develop into thalamus. So we are quite curious which signalings will be inhibited by this physical boundary. So we just find the one member in Wnt family called Wnt7b. It seems that aggregate only in P3 region, and all other members of Wnt family located in ZI region here or in P2 regions. So we use the cell-cell interaction to study the Wnt7b and the downstream TFs and target genes that have the interaction with Wnt7b. So this is the anterior-posterior analysis. And we also curious that the ZLI is like a ring insert into the brain. Remember that the brain is a hollow tube in this stage, and this is like a ring, ZLI like a ring insert into the brain. However, the ring is not closed. You can see here, the ring is not closed. So what are the other genes expressed here? We call that the root plate to inhibit the ZLI, and finally the ZLI cannot form into a closed ring. So we can also analyze the gradient genes along the dorsal to ventral axis, and we found that FGF8, this is already known morphogene, inhibited SHH expression, and the downstream target gene seems like SUFH that exactly expressed and interact with FGF8 to inhibit the expression of SHH so that the ring cannot be enclosed. Finally, I will introduce a cross-time analysis in our spatial toolkit. The non-biological event is for the Drosophila development. The midgut is original divided into two parts, either in anterior or posterior. During the development, this midgut fused into one organ. However, this is not the end of the development. The midgut will be twisted so that the food will stay in the midgut for longer time for digestion. So we just calculate another field. Notice that I have already introduced the potential field. The perpendicular field of the potential field is vector field. I can easily calculate the vector field to simulate the cell migration here. The cell migration across two developmental stages. We can see they not only the cells fused together, they also twisted. The twisted is called torsion in differential geometry. It is like winning out the tower to get such force in morphogenesis. And we also calculate all the genes in this vector field to see which genes are expressed correlated to this movement of this cell migration. So we find some novel genes that correlated, or after experiment, we can prove these genes will guide this morphogenesis during the midgut development. So we also demonstrate this algorithm in the heart development. As we know, heart have 4 chambers, while the left ventricle, this chamber, is derived from first heart field in early stage, and the other 3 chambers are mostly derived from the second heart field. So when we calculate all the differential geometries, we find either in acceleration, divergence, or Q, the left ventricle is most conserved, while other 3 chambers, as well as the outflow tract, have different features in these geometric calculations. We also find the genes that have highly correlation with each geometry. And these genes, most of genes, have been experimentally verified in mouse analysis. At last, we also generate an interactive website called Spatio to calculate all these algorithms by mouse clicked in this website. So Spatio is a user-friendly tool for us if you are not quite familiar with the algorithms that I introduced before. So all this work with collaborate with Xiaojie, and I also acknowledge the help from Jieping and Zhao Rong from Ali Calculation. Thank you.